Hello, my wonderful 7th graders, and welcome to a new episode of What Are We Doing in Class Today? Today's episode is all about online microscopes. So we're going to be working with a virtual microscope to figure out basically how to use a microscope and how to see things that we're not able to see with our human eye. Hopefully in the next coming years, I will have the materials to be able to do some different things with you guys to show you guys some of these things we might see in a microbiome or that are too small to see. But for right now, we're going to learn the same kind of concepts with this virtual microscope. So your learning targets, I can practice the basics of using a microscope virtually. Okay? All right, so the first thing you guys want to do is obviously learn a little bit about microscopes. So you guys are going to right-click this handy-dandy link right here and open it up, and it's going to take you into this Amoeba Sisters YouTube video. So you guys can go ahead and watch it. It's about 9 minutes long, but it'll basically talk about all the different parts of a microscope, different types of microscopes. So most of the pictures that we've seen in class are what we call a scanning electron microscope, where we get to see really cool images of the surfaces of cells, okay? And so again, if you look back at a lot of the backgrounds that we've had from our past assignments, those are all scanning electron microscopes. So you guys are gonna learn a little bit of a difference about that versus a transmission electron microscope, which is also a really fancy piece of equipment, but you see more of the inside of a bacteria or something really small, and even viruses. And what we use in class, just to see basic cells, is going to be a light microscope. So you guys will learn how to use a light microscope through this video, and then also with the virtual link. Okay, so once you guys have finished the video, you guys can restart this video, and then we'll move on to the next slide. So make sure you pause to watch that. Okay. All right, moving on to slide 3, here is your link for the virtual microscope. So you'll want to go ahead and right-click and open that link up, and it's going to take you into a page that looks like this. We're going to go ahead and launch our activity. So there is a series, and it might take a second to load, but there's a series of slides that we're going to be looking at. But just to get the basics down, we're going to look at the letter E. So let's take a look at what this looks like in your experiment or in your guys' assignment, okay? So here's how you use the light microscope. I've already showed you to launch the activity, and then once it loads, you guys are going to click Explore. We're almost loaded. Okay, and then to get the letter E, you guys are going to click on the slide box here. You're going to click on Sample Slides, and then you're going to find the letter E. If we were to look at a microscope prepared for this, or a microscope slide that was prepared for this lab in class, it would look like this. We would take a little letter E out of a newspaper. We would drop it on this glass slide. We'd put a drop of water on it, and then we'd gently take a slide or a slip here, and we'd drop it over top of that letter E to hold it down. Okay, so that is what it would look like prepared. This is what you guys would stick on the microscope. Okay? All right, so we're going to go ahead and see that right here. We're going to click Explore right here. We're going to choose the slide box, the question mark, click right on it, and click Sample Slides, and choose the letter E. So what you guys will see now is if you were to look through the microscope, this is what your view is going to look like. Obviously, it's a little bit blurry. The letter E is not microscopic, so you guys are seeing it barely magnified. It's only a 4 times magnification, so you're seeing it 4 times closer. And you guys will see that it's pretty clear to see almost right now. So I'm just going to show you guys how to work through that after I show you guys the instructions. So to get this focused, you guys always want to start with the lowest magnification and get it focused there first. Okay? So before you guys go to your higher magnifications, you focused every step in between. So before we go to anything higher, we're going to get our 4x magnification focused first. So first we're going to start with the coarse focus knob until the E is somewhat clear. Then we're going to do the fine focus knob until we can basically get it as clear as possible. And then if we need to, you guys can kind of adjust the light and see what that does. So again, we're going to start, we're on 4x. Okay, we are going to start with our coarse focus and drag that and get it as clear as we possibly can. Normally this would be a knob. And then here's our fine focus. Because we're only on the letter E, it's something that it's visible to the human eye. We don't really have to do a whole lot of fine focus, okay? And then you guys can kind of mess around with the light adjust to see what the light adjust does for you. Brighter doesn't always mean better, okay? So you guys can see that letter E pretty clear. If you guys look back This is what the slide looked like before you put it in. So let's kind of examine what it looks like after. Okay, here's the last little reminder for you is you need to screenshot this 4x magnification of your letter E. So here's your guys' reminder on how to screenshot. Okay, remember if you're using a computer, you're going to hold down the control and the shift buttons, and then you're going to hit this little key which is located right above the number 6. If you guys are using something other than a Chromebook, you guys can use the snipping tool. So if you're using one of my computers or a different computer at home, search for your snipping tool and then click New. Click and drag what you guys want to copy. It should just be this little part right here that has the letter E. And then copy is Control C and paste is Control V. You guys will want to paste that in right here. Okay? And then answer this question. What two things happened to the orientation of the letter E when it was placed on the microscope? Again, compare it to slide 6. So if I look back, this is what it looked like before. Okay? This is what it looked like after. There's two things that happened to how that letter E is oriented. There's one that's obvious and one that might be a little bit harder for you guys to figure out. Okay, it's obviously bigger. That's not an answer. This is orientation. Okay, so think about what happened to that letter E. It wouldn't read the same way if you were to read that letter E in a newspaper. Okay, so answer that question here. What were those two changes you saw? Bigger is not one of them. Okay, all right, moving on. You guys will click remove slide to get rid of the letter E. And then you guys are going to go on to the onion root tip cells. So if we were to take an onion plant and look at the cells found in the root tip of that. Okay, so if we looked at an onion root and then just looked at the very end of that root, that's the onion root tip. Okay, so you're going to go to the slide box, plant slides, and then look at onion root. Plant slide, onion root. And that's going to stick under the microscope for you guys. Just like with last time, you guys are going to want to focus it in first on your 4X. So always start with your coarse focus until you guys get it about as clear as you can. And start with your fine focus and get it as clear as you can. Obviously, this is an onion root. Okay, this is something that's visible to our naked eye, but the cells in that onion root aren't. And that's really what we're looking for here. Once you have it focused in 4X, then you may move to your 10X. Okay, if you look at your next slide, which is your instructions, you guys need to screenshot all of these 3. So here's your 4X. So you'd take a screenshot of this, okay? And then you'd move on to your 10X. So I'm going to go ahead and click on 10X. Notice that it's not going to be super well focused, so you guys might need to do a little fine focus until you really see those lines well. Okay, get it focused here. Take a screenshot of that. You guys are going to put that right here at 10X. And then you guys will move up to 40X. Okay, same thing there. You guys can kind of mess with your light. Maybe your light adjust makes it a little bit better. And then focus it as best as you guys can. Once you think you have it focused in good, okay, then you can take a screenshot of that and put it in the 40X box. Okay, oh, X box, All right, your questions for this. Each little box you see is a plant cell. So here is a little square. That is one cell in this onion root tip, okay? Inside that little box, you see a dark round circle. That's the nucleus. The job of the nucleus is to hold all of our genetic material, or our DNA in the case of an onion root tip cell. Okay, so in here, this dark little circle, we find the DNA. So what you guys are going to do is you are going to go back to the last slide and you are going to label one cell and one nucleus. Okay, so you should have a picture right here under 40X. Use your draw tool to label one cell and one nucleus. Okay, so maybe trace outside of the box here for the cell and then use a different color to trace the nucleus and then write nucleus and cell. Okay, as best as you guys can. Then answer this question. What do you notice about the shape and arrangement of these cells? Okay, so click to type right there and answer that question. Then we're going to move on to human cheek cells, or the best rendition of human cheek cells that this slide box has to offer. So you guys again will click on the slides, you will go to human, and then closest thing to human cheek cells we have are what we call simple squamous epithelium, which is also very similar to your outer layer of skin on like your hand or your arm or anywhere around your body. Okay, these are basically one thin layer of cells. When you guys pull this up, you won't really notice a whole lot of anything. You really have to scroll for this one. Here's my simple squamous. Okay, remember to focus as best as you can under your 4x first. Use your coarse focus first, and then your fine focus, and then screenshot that. Then you can go to your 10x. Okay, use your coarse focus first. Get it as best you can, then use your fine focus to really fine touch it up. You guys can mess with your light adjust. Okay, and once you guys have it focused there as best you can, go into your 40x. You guys should screenshot each of those as well. So 4, 10, and 40. Okay, and then if you go to 40, again, you guys can kind of use your fine focus to focus. The dark little circles, so these are kind of hard to see. One thing that's kind of cool about this is you guys can click to drag. So if we, let me find you guys a really good cell to look at. These are all good cells. So the darker circles are the nucleus. So here's me just moving my slide around. I just want to find one that has a good border for you guys to see. I'll give it a little bit more light. A little bit more focus. These ones are a little bit different. So what you guys will notice with this is that you don't have quite the distinctive edge around your cells like you do with a plant cell. Okay, they are not regularly shaped. Okay, they kind of have like a weird circular shape, whereas a plant cell has this little box shape. So that's a big difference between plant cells and animal cells. Gosh, I just want to find one good cell that I can show you guys beautifully. So here's kind of one right here. You guys can kind of see the outside border of that. Okay, here's one. You can kind of see the outside border of that. They're not shaped like a square. They're kind of just all mashed together. Okay, and then again, that little dark area in the middle is your nucleus. Okay, so you guys will go ahead. You'll do the same procedure as you have on the last ones. You guys will do your 4, your 10, and your 40x. And then you'll answer this question. What do you notice about the shape and arrangement of these cells? I've kind of already talked to you guys about that. And then your last little one that you guys are going to do based on my directions is the bacterial cells. So as we know, bacterial cells are way smaller than human or animal cells or plant cells like we've seen already so far. So you guys are going to go clear up to 100x. So you guys are going to click on your box. Oops, you're not going to do plant slides. You guys are going to do bacterial slides. And you guys are going to do the gram stain. Okay. Once you guys get that, you guys are going to get it on 40. You guys will see, or excuse me, 4x. You guys will see a little focus button on the 4x. You'll see like barely any purple dots when you guys get it focused in on the 4x. So do the best you can to get it focused there, and then move to your 10. Okay, and get it focused there as best as you can. You guys might want to mess with your light adjust a little bit more on this one too. So focus that in as best you can. It's kind of hard because you don't really know what you're seeing yet. Okay, and then once you have it focused as best you can there, go to your 40. Okay, and you can kind of move it around a little bit. You can mess with your coarse a little bit, your fine a little bit, and then you guys can mess with your light adjust a little bit. And then when you do 100, it's going to ask you to do oil immersion. So if you notice this little question mark here, when you guys go clear to 100, it's really hard to take the light through that kind of magnification, so you actually have to put a drop of oil on your slide. So that's what we're going to do right here, is add this drop of oil. And then what we're going to be able to see is these little bacteria cells are these purple. So anytime we hear something that's gram stained, it's stained purple. Okay, it's looking for something specific on the outside of the bacterial cell, and it turns it purple when we stain it. So that little guy right there, there's 3 little bacterial cells there. I think like 2 there. It looks like there's about 4 right there. So they're clustered together really close to each other. Okay, that looks like a single one maybe right there. Okay, you guys will get, for this one, screenshots of your 10, your 40, and your 100. So I don't need a 4x on that one because you really can't see a lot with your 4x. And then answer this question. For the bacterial cells, you should have gone clear up to 100 times magnification. What does this tell you about the size of bacteria compared to human or animal cells and plant onion cells, basically the cells we've looked at prior to this? Okay, so what does that tell you about how big the size of bacteria cells are compared to those animal and plant cells? Okay, finally, you guys get to do a little free explore. You guys can choose one other slide of your own. Start with your 4x magnification and go clear up to 40. You guys only need to screenshot your 40 Tell me what slide you chose here, and then tell me just what you kind of noticed about these cells, okay? Just give me some kind of observation of what you saw. Then complete your wrap-up. Circle, do you have any questions, yes or no? Remember, you don't have to make up a question for this. It's only if you guys are confused about something or you have a question to ask me in general. And then 4, 3, 2, 1, how do you think you did on this assignment? Magnificent job, but don't be a water bear and forget to hand it in. That's a water bear. He's a microscopic organism who can live through just about anything. Anyway, I hope you guys are having a great day, and I'll talk to you soon. Bye.